Today we will be looking at nucleic acid probe labeling. Note was prepared by Dr. Stephen Amoa, my lecturer at Kwame Nkrumah University of Science and Technology, Faculty of Agriculture, Department of Crop and Soil Science. Nucleic acid probe labeling simply means development of probes. And a probe is a short sequence of DNA or RNA that is used to detect DNA or RNA molecules. Many of the techniques of gene manipulation depend on the hybridization of nucleic acid probes to a target DNA or RNA sequence. This means many of these techniques of gene manipulation will depend on the development of probes. For instance, we have two varieties of cowpea. One is resistant to, let's say, droughts, and the other one is susceptible to drought. But we want to find out whether the one susceptible to drought, at first we didn't know whether it was susceptible to drought or not. So we would develop a DNA or nucleic acid probe and we will use it to detect whether that gene which is responsible for drought in A is also present or absent in B. So we will develop these nucleic acid probes to do that. So they are basically used for detecting DNA or RNA molecules. So here are few of the applications. Detection of DNA or RNA molecules can also be used in situ hybridization, also used in the detection of microorganisms in clinical specimen, also used in DNA profiling, that's DNA fingerprinting. And here are some of the methods that we use in developing nucleic acid probes. We have 1. Nick translation, 2. Primer extension, 3. Methods based on RNA polymerase, 4. End labeling, and then 5. Direct labeling methods. And today we'll be looking at 1 and 2, Nick translation and primer extension. With the Nick translation does not have any relationship to do with translation itself. As translation, you know, it is the synthesis of protein. But here, Nick means to make a small cut, okay, on a DNA molecule by very limited treatment with denase 1. So what this means is, let's look at this. So for Nick, as I said, is a small cut. So we would make a small cut here. This one is a Nick. We've made a cut here. So we've come to make another cut here. We've made another cut here. Okay, this is Nick. But it doesn't mean this. If it goes through this way, this one is not Nick. This one is not Nick. But for this one here, this is called Nick. This cut here is Nick. This cut here is Nick. So let's extend our line. Okay. And let's get back to our notes. So DNA polymerase 1 is then used to incorporate radiolabeled deoxynucleoside triphosphate, that is 32P, and then DNTP. So let's get back to our drawing here. So the RNA polymerase would now be used to join this one, the appropriate one. If this one is G, here it will be C. So we will label the nucleotide. We will label them so that when the polymerase inserts it here or use it to join the sequence, and we view it under special equipment, we'll be able to see the radiation being given out, the light or emission being given out, so that we'll be able to detect that yes, we have this particular sequence here, we have this particular nucleotide here, we have this particular nucleotide here. So that is what they are saying. And the DNA sequence remain unchanged. It doesn't change. It is what is missing here that the polymerase will bring it back. So if here is G, here will be C. If here is T, here will be A. In that order. In that order. So basically that is how Nick translation labeling is done. We will have our Nicks, that's our small Cas. After that, we would have our polymerase extending our Cats or the Nicks. So most often the reaction is performed with only one of the 4 deoxynucleoside triphosphate. So it's only one that we label. If it is C, we are labeling only C. So wherever the enzyme has made the Nicks, let's say, let me show you what I mean. So the enzyme has make Nick here, so meaning here is C, here will be C, here will be C. So we label only C, our nucleotide C, and the rest you will not label them. A, G, T, we will not label them, but we label only the C so that it will incorporate our C in the appropriate region. Because the Nick process occurs at random, a DNA preparation is effectively labeled uniform. Yes, and a major advantage of Nick translation over other uniform labeling method is flexibility with respect to probe size, specific activity and concentration. Label probe. Okay, let's see what we mean by all what I have said in the slides. Okay, so at first duplex DNA is denatured. So when we denature the DNA, we have our single-stranded DNA here, and the primer, the random sequence primers are added to make a label copy. So now when the primers are added, they will initiate the extension, and the polymerase enzyme will extend to make a complementary copy. So it will complement it. So for instance, let's say we have the C. Okay, C. So here is the C. Yes, C. So this side we will have our C there. Okay. So our C will be incorporated here by the enzyme because it is labeled. It is only the C that is labeled. The rest of the nucleotides are not labeled. We have labeled only the C. So this is what we would have. So first of all, it's a double-stranded DNA. We will denature it to become a single-stranded, and we will make a complementary copy of it. So this green color is a complementary copy, and we would have all the nucleotides to be the normal nucleotides except the C, which is radioactively labeled, and the primers will be added to the sequence, and the polymerase will do the extension for us. And wherever we get, and we have G, if you have G here, the opposite one will be C. So the enzyme will fix the radioactively labeled C there, and after that we denature it again. We denature this double-stranded to release our probe labeled. So this one is now our probe. You see, we have the C here, so it is now labeled. This is the whole process of the primer extension. At first we would denature our double-stranded DNA to form our single-stranded DNA, and we would make a complementary pair of it, and we denature it to release our now labeled probe. We would now use this to detect in another solution whether a particular gene of interest is present or absent. We need a specific region to be detected. This is the one that you should prefer, the primer extension method of designing of probes. So you design your probe based on this method, and you would actually be using it for a high specificity and low probe concentration. We don't need a lot of probes concentration in our sequence. If you haven't subscribed, kindly subscribe and click on the notification button for more of my new videos.